Our next speaker is going to be Banu Rekepalli from NICS, talking about some work on Kraken. And I don't have the PI up in front of me, Banu, but hopefully you'll remind us who you are working with when you get going on the talk.
Yeah, will do.
Yeah. Thanks very much. Go ahead and take it away.
Okay, this is more like just one science use case rather than many, like what Syed is working on. This is docking studies with Professor Yuri Peterson from Medical University of South Carolina. Can everybody hear me okay?
Just a little quiet, but not too bad.
Okay. Maybe this is better. Like when I first presented at MUSC, they were doing these docking studies on small cluster they have there, and it's taking about 3 months of flexible docking time per study. Like they're using 1.5 million compounds to dock onto a protein from ovarian cancer pathways. And that's where they started their work on Kraken. Could we go on to the next slide, please? So I will briefly talk about some of the studies that we have done, and we built a wrapper rather than changing the Doc6 code, and we look at some results and future work. Next slide, please. To briefly talk about docking, so you have a protein receptor, so it's a 3-dimensional structure, and you're trying to dock these small molecules onto it, where you can see figure A is a protein and B is they're trying to find the appropriate site where they can dock these small molecules, and they're building these spears, and also they put a grid around it so you can dock different kinds of molecules on this. So the database we are working on is Zinc database. It's a curated database by the PI. It has 1.5 million small molecule compounds. And PI is mostly interested in docking to the proteins related to ovarian cancer. Next slide, please. Initially, as I said, when they were doing the studies at MUSC, it took about 3 months to run one set of like 1.5 million compounds onto their protein. And like we took their application, put it on Kraken, even single instance of Kraken, single instance of Doc6 run on Kraken was about 20x faster than what they were running on their small cluster, which is the older cluster at that time. We took these Doc6 program, we looked at it. Next slide, please. We profiled the code using gprof and some print statements, and looked at various communication patterns using CRAPAT. They had an MPI version, but it writes after every completion of every compound docking, and it was boggling up the bandwidth as we go higher and higher. So we decided to move away from the existing version they had, and then we wrote our own wrapper. Next slide, please. So we call this HSPwrap, which stands for Highly Scalable Parallel Wrap, and it has like master, a controller, and a worker. So it's a distributed MPI program, and we wrote one wrapper to solve most of the data parallel applications. This is also used for other bioinformatics applications and some other informatics applications, but today I will be talking mostly on Doc6. We look at different ways to do dynamic load balancing and how efficiently we can optimize I/O and few fault tolerance functions were added to this. And this code is basically reusable on different kinds of architectures. Next slide, please. So this is the basic structure of our wrapper. So we have a master and controller and worker is on every worker node. So master is one node and controller worker on every node. So the master is responsible for moving both databases and query sequences, query blocks from the file system to all the worker nodes. So we basically get everything once and we send it to all the worker nodes. Next slide, please. So basically, yeah, master broadcasts the database to every node, and like the broadcasting was scaling logarithmically, so it was scalable. And also master distributes the compressed blocks for the input query to reduce, again, I/O. And next slide, please. So we have a controller on every node. And there's only one rank per node basis, and we use threads to spread work around. So this controller is responsible for getting work from master and also writing to the file system. So every time a worker process, so we on Kraken, as everybody knows, it's like 12 cores, so we have 12 processes running doc6, and we keep them always busy. So we have a queue for the input. So once the queue is halfway emptied by the workers, we keep on filling the queue, and the controller is responsible for filling the queue, and the worker threads just keep on crunching and writing the results to buffers. So we have 2 buffers to which we write to so that the worker keeps on working. Next slide, please. Okay. So as I said, we just wrote this Stdio Wrap to actually wrap the code itself, and we do not change any functionality of doc6 program. So this replaces the standard I/O functions when linked with the target programs, and also it uses like shared memory segments. So there's only one database, and all 12 threads sees the database, and also it writes to the buffers and offloads expensive operations to optimize implementations. Next slide, please. So we keep track of how many queries are done in this. We do basically check some kind of thing. So see how many input queries are done, and we keep track of them, and we properly formed blocks can be omitted. And whenever a specific job fails, we can go see how many have done, and we can resume from failed spot. So we can just take only those queries which do not go through, and we rerun them in a different job. Next slide. So the input data, as I said, it was 1.5 million compounds. These are all curated from the Zinc database and few from PubChem database. And PI was interested in docking on 8 proteins related to ovarian cancer pathway. And he wanted about 5 conformations per protein. So if he has done this 40 different runs on his cluster, it takes about 120 months, uninterrupted time on his cluster. So we were able to run these using jobs anywhere from 1,000 cores to 4,000 cores. We didn't do much of the scaling study yet. We are in the process of writing a publication. So we're generating results for the PI because he needs these results for some of his publications. So each run generates about 200 gigabytes to about half a terabyte of data, output data, and roughly about terabyte to 2 terabytes of data per experiment set. That means about 4 or 5 conformations per each protein. And once you parse this data, just get the values that PI is interested in, it gets down to about 200 MB per experiment. And it took about 300,000 SUs for running 25 conformations for 5 different proteins. Next slide, please. So this is pretty much what the results represent. PI plots this, and he has a cutoff of -20 e power -25 or something like that, and he gets the compounds which falls below that. There are about 400 or 500 compounds that he's interested in. Next slide, please. And yeah, this shows like total number of successful compounds per target. So this shows 4 targets, about 5 conformations, so 5 runs, and like he gets hits about 300-400 compounds that he's interested in from every run. These are done redundantly so that he can make sure that the compounds are hit. Next slide, please. So yeah, so that's pretty much what we have done for the PI. And we are also trying to build the downstream analysis. So to take the terabyte of data, right now he moves every time an experimental set is done, he moves the entire data to their clusters and run the analysis on their clusters, which takes about a week or more to do the analysis per experimental set. So we are trying to take all the tools that they are using and run them on HPC resources here. And we also are interested in porting Autodoc and Rosetta and run the same experiment again. So that he wants to see whether the other tools will have identical hits as Doc6 does, so that he can reduce false positives. And he's also looking at various tools. So this is done to reduce the compound space. And we are also interested in porting this to a science gateway so that other people can use the same parallel version. So that's pretty much what I'm working with the PI on this project. I guess that's all I have.
Banu, terrific. Thank you very much. Questions from anyone on the line for Banu?
Hi, Banu. This is Sudhakar again.
Yeah.
So I have a couple of questions. One is, so you mentioned that you are actually loading the Zinc database, you're distributing the Zinc database onto multiple processors.
Yeah, so...
Right. I mean, so the question is, so can you not simply, it's a huge file, right? It's a 1.5 million compound, probably growing. maybe after curation, it's probably not a very big file, but you can keep it, but then you can use some kind of a parallel I/O to read the database, because I think you're using structures essentially independently, right?
Yeah, so basically master, it divides the work. So we load everything to the master node, and master node sends like just a random number. So it sends about 24 compounds per node, and 12 processes are running on one of the compounds. And these 24 compounds are kept in a queue, and then once like 12 processes take the 12 compounds, that queue is refilled. So we're not sending the entire 1.5 million compounds to all the nodes. We only we divide the compound database and send the jobs to every node, about multiples of 12, like 4 or 5 multiples of 12. That's about it.
Banu, thanks. I was interested when I first heard about this work at how you leave the internals of the codes the same but wrap externally, and how very much that allowed you to scale, I think in one of the other presentations that I saw, and how many different codes you've done this for. Are you, when you don't change the internals like that, are you essentially running lots of independent doc6s in this case simultaneously? Yep.
Yeah. So it's basically just the binary of the doc6 is run on pretty much every core.
Right, and then the I/O is a bit optimized because of the things that you do also, right? And
that's right. So we have buffering techniques. So we put everything in a buffer when every core finishes a job. It goes straight into the buffer, and we have multiple buffers. So we take the results from one buffer, compress it, and put it in the other buffer. And once this compressed results are like full, just certain percentage, we flush it out to the file system. And also we have hierarchical directories so that like we can use parallel I/O at that point.
This is Kim Dillman.
Oh, yeah, go ahead, Kim, please.
Well, I was just going to ask, because from the descriptions of it, sounds like what you're doing, it's almost like that this is, you kind of wrote like a workflow manager for high throughput type code, because I know I've Rosetta and Autodoc, both of those codes, various PIs have run them on the Condor pool at Purdue. And so that was one of the questions. It sounds like this is basically kind of your parallel wrappers, kind of like putting a workflow manager to lots of serial jobs. Is that a good description of it, I guess?
Yeah, it's like we can call it as ideally parallel or embarrassingly parallel
right.
workflow, yes.
Yeah, so what you're doing is basically managing the distribution. So it's like a workflow thing, because what you wrote is just managing the distribution of all these independent jobs and then collecting the results back in the end. And
yes.
About how long do the individual jobs usually take? I mean, are they long jobs, or they don't take very long? Or
they vary about like the compound to compound. So it's anywhere from few seconds to few minutes.
Okay, so basically it is a very, and that's why probably I've seen Autodoc and Rosetta at least on the Condor pool, and I think one of those maybe even on OSG. So that answers my question, because I kind of thought that's what you were describing, was that all these things were independent, somewhat parallel, independent of each other. You know, but the parallel part was really more of the managing the distribution and collecting the information at the end.
Yes.
Okay.
Yeah. So Banu, this Suresh, you mentioned about the Science Gateway plan. So is there a plan to adopt, bring this tool to an existing Science Gateway you're working, or are the PIs thinking about starting a new Science Gateway?
We are still in talks with PI. So PI and I, we are working on different grant proposals. So we want to provide this docking as a service. So we also want to don't want to do redundant work. So we will store everything in the database. So it might be a separate gateway by itself if it gets funded. but it could also be added to the systems biology gateway that we have currently. So it all depends on the PI.
Sure. Yeah. Thank you.
Banu, thanks very much. It seems like a nice general purpose solution, and I think the optimization of the I/O is kind of a nice extra feature that probably a lot of the typical workflow packages don't have also. So very, very nice work. I know we're over time. Thank you to everyone for staying. Thanks to Banu and Syed for terrific presentations. And hopefully I'll get the AT conference invalid credentials thing sorted out by next week or next month. So sorry about that, folks. Thank you very much.
Thank you, Nancy.
Yep.
Thank you.
Thank you.